At low temperatures, microbes capable of decomposing plastic have been discovered by scientists in both the Arctic and the Alps. The discovery, development and genetic modification of organisms capable of breaking down plastic waste not only contributes to the reduction of pollution, but it has also become a lucrative industry. While a few microorganisms have been identified as having the ability to perform this task, the enzymes that enable their plastic degrading abilities when applied to an industrial scale generally function only above 30 degrees Celsius. As of now, the cost of industrial applications remains high due to the substantial amount of heating they require, and they are not environmentally friendly. Nevertheless, there may be a viable solution to this challenge. By seeking out specific microbes that are adapted to colder environments, it may be possible to find enzymes that function at lower temperatures and thus reduce the need for excessive heating. The scientists from the Swiss Federal Institute WSL conducted a study to identify microorganisms in specific regions with different climates. They focused on high altitudes in the Alps of their country and the polar regions. The results of their research were published in Frontiers in Microbiology. Dr. Joel Ruthie, the first author, who is currently a guest scientist at WSL, has revealed that new microbial groups found in the plastisphere of Arctic and alpine soils have the ability to decompose biodegradable plastics at temperatures as low as 15 degrees C. As a result, these microorganisms could potentially decrease the costs and environmental impact of enzymatic methods for recycling plastic. The study conducted by Ruthie and team involved the examination of 19 strains of bacteria and 15 strains of fungi. These strains were found growing on plastic waste that was either intentionally buried or found free on the ground. The plastic litter from Svalbard was obtained during the Swiss Arctic Project 2018, which was aimed at providing students with a first-hand experience of the effects of climate change. In addition, soil samples from Switzerland were collected from the Muot da Barba Paida summit, which is 2,979 m high, as well as the Val Laviren Valley in the Canton Graubünden. The laboratory scientists allowed the microbes to grow in isolation as single-strain cultures in an environment devoid of light and kept at 15 degrees C. Through the use of molecular techniques, the scientists were able to identify the microbes. The results displayed that the bacterial strains were members of 13 different genera in the phyla Actinobacteria and Proteobacteria. The fungi strains were members of 10 different genera in the phyla Ascomycota and Mucoromycota. Using a series of tests, they screened each strain to determine its capacity for breaking down sterilized samples of non-biodegradable polyethylene, PE, and the biodegradable polyester polyurethane, PUR, as well as 2 biodegradable mixtures of polybutylene adipate terephthalate, PBAT, and polylactic acid, PLA, that are available on the market. After 126 days of incubation, none of the strains were capable of digesting PE. However, 19 of the strains managed to digest PUR at a temperature of 15 degrees C, which accounts for 56% of the total strains. Of these strains, 11 were fungi and 8 were bacteria. Furthermore, 14 fungi and 3 bacteria were able to digest the plastic mixtures of PBAT and PLA. Nuclear magnetic resonance, NMR, and a fluorescence-based assay were utilized to confirm that these strains were able to break down the PBAT and PLA polymers into smaller molecules. According to Ruthie, the discovery that a significant portion of the tested strains had the ability to break down at least one of the assessed plastics was unexpected. 2 fungal species of Neodivresia and Lachnalula were the most proficient performers in the study. These species had the capacity to decompose all of the analyzed plastic, with the exception of PE. Furthermore, the study revealed that the ability to decompose plastic varied among strains, depending on the culture medium that was used. In fact, each of the 4 media tested elicited different reactions from each strain. The evolution of the ability to digest plastic is an interesting topic to consider. Due to the fact that plastics have only been in existence since the 1950s, it is highly unlikely that the ability to degrade plastic was a trait specifically targeted by natural selection. Dr. Beat Frey, a senior scientist and group leader at WSL, explained that a diverse range of polymer-degrading enzymes that aid in the breakdown of plant cell walls have been discovered to be produced by microbes. It has been observed that plant pathogenic fungi are especially skilled at biodegrading polyesters due to their capacity to produce cutinases, which are capable of targeting plastic polymers as they bear a resemblance to the plant polymer. Cutin. The successful strain's enzymatic activity at the most favorable temperature remains unknown, as Ruthi et al. Exclusively analyzed digestion at 15 degrees Celsius. Frey stated, "It is common knowledge that the majority of strains that were tested have the ability to thrive in a range of temperatures, specifically between 4 degrees C and 20 degrees C, with an optimal temperature of approximately 15 degrees C. A significant obstacle that must be overcome is the identification of the enzymes that degrade plastic, which are produced by microbial strains. Once identified, it is necessary to refine the process in order to produce substantial quantities of these proteins. Moreover, it may be necessary to alter the enzymes to further enhance their properties, including protein stability. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.